Hello everyone. We learnt about how the zygote divides and differentiates to form the embryo in the last module. The developmental processes leading to embryo maturation and seed formation start after the morphogenesis phase of embryo development, which is completed by the heart-shaped embryo stage. In this module, we shall study these developmental processes which lead to embryo maturation, endosperm development, and the formation of the seed. The plant hormone abscisic acid plays an important role during the maturation phase of the embryo. The learning objectives in this module are understanding the processes that take place during embryo maturation, the role played by the hormone abscisic acid in these processes, and the development of the endosperm and its regulation during seed formation. Let us start now. The basic body plan of the embryo is established by the heart-shaped stage of embryogenesis as seen in the earlier module. Then the embryo grows by cell expansion and shows an embryo axis consisting of the root and shoot apical meristems, which are separated by the hypocotyle. It also shows two cotyledons which enclose the shoot apical meristem. Besides zygote formation and embryogenesis, the fertilized central cell develops to form the endosperm. Accumulation of storage products occurs in the endosperm cells. In some species, the endosperm degrades as the embryo grows, and the cotyledons serve as the seed storage organs. The storage products may be proteins, starch, and lipids, which support the growth and development of the seed during germination. This is called the seed filling stage. Besides storage products, the embryo also accumulates specific molecules like oligosaccharides, late embryogenesis abundant proteins or the Lea proteins, and the heat shock proteins that protect it against the desiccation stress which sets in as the seed loses moisture and reaches the end of the maturation phase to enter a dormancy phase. The dry seed represents a pause in the plant's life cycle, which can withstand unfavorable conditions in the environment. Before proceeding to the development of the seed, let us recall that seeds are the product of two independent fertilization events that give rise to the diploid embryo from the fertilized egg cell and a triploid endosperm from the fertilized central cell. The embryo and endosperm are genetically similar in that they have representation of both the maternal and paternal genomes. However, the embryo shows the representation of the maternal and paternal genomes in the ratio of 1:1, whereas the endosperm shows the representation of the maternal genome, which is twice that of the paternal genome, because the central cell is diploid and it fuses with the haploid sperm to form the triploid endosperm. The seed has a coat which is entirely of maternal origin, which is formed by the integuments of the ovule. Now the endosperm arises from the fertilized central cell. First, the central cell nucleus undergoes a series of mitotic divisions to produce a multinucleate expanding central cell that surrounds the embryo. Then at the hard stage of embryogenesis, cellularization of the central cell begins to form an endosperm tissue consisting of large specialized transfer cells that facilitate nutrient transfer from the maternal tissue to the developing embryo. So the seed is made up of maternal tissues that form the ovule and the tissues formed after fertilization, namely the embryo and the endosperm. The plant hormone abscisic acid plays an important role during the process of seed maturation and also in the induction of dormancy in the mature seed. Abscisic acid biosynthesis starts from the carotenoid beta carotene in the plastids. Beta carotene is converted in a series of steps to the xanthophyll neoxanthin. Neoxanthin is then acted upon by an enzyme 9-cis epoxycarotenoid dioxygenase or NCED, which converts neoxanthin to xanthoxin. This step marks the first committed step towards abscisic acid biosynthesis. The xanthoxin then moves out of the plastids and is converted in two steps to abscisic aldehyde and finally to abscisic acid. Now, this bioactive form of abscisic acid is not the only form in which abscisic acid levels in the cell are regulated, but abscisic acid levels may also be regulated by its catabolism. Enzymes belonging to the cytochrome P450 monooxygenases called CYP707A is responsible for hydroxylating the bioactive abscisic acid to form a catabolic product 8-hydroxyabscisic acid. So NCED and CYP707A are important enzymes which regulate the levels of abscisic acid in a cell. Now, besides regulating the levels of abscisic acid, perception and transduction of the ABA signal are also important ways of regulating ABA effects. Abscisic acid is perceived by a receptor which is called as the pyrobactin resistance or pyrobactin resistance like or regulatory components of the ABA receptor. In short, it is called as the PIR-PIL-RCAR receptor. In the absence of abscisic acid, the transcription factors, which are the ABA response element binding factors or ABFs, cannot bind to the promoters of ABA inducible genes because they are not phosphorylated. They need to be phosphorylated by enzymes which are kinases, which are belonging to the family of sucrose non-fermenting 1 related kinases. So in the absence of ABA, the SNRKs are not phosphorylated, and because there is an enzyme called as protein phosphatase which prevents or removes the phosphate group from these kinases. In turn, the SNRKs are unable to phosphorylate the ABFs. Now, when abscisic acid is present, the abscisic acid bound to its receptor attaches to the protein phosphatases and prevents them from removing the phosphate group from the SNRKs. This allows the SNRKs to transfer the phosphate group to the abscisic acid response element binding factors or ABFs, and these ABFs can then bind to the abscisic acid response elements present in the promoter region of abscisic acid inducible genes to carry out their transcription. Abscisic acid signaling is known to activate a network of transcription factors which are active during the process of seed maturation. This network is called as the LAFL network. It includes 4 transcription factors called leafy cotyledon 1 or the LEC1, abscisic acid insensitive 3 or the ABI3, FUSCA3 or the FUS3, and the leafy cotyledon 2, LEC2. So the first letters of these 4 transcription factors are used in coining the term LAFL network. ABI3 is a transcription factor that is necessary for the expression of a large number of late embryogenesis abundant genes, which are thought to be essential for the acquisition of desiccation tolerance. ABI3, along with the B3 domain transcription factors LEC2 and FUSCA3, and another transcription factor, LEC1, work together in regulating various processes during seed formation and dormancy induction. FUS3 and LEC1 regulate the accumulation of ABI3 protein in turn, and LEC1 functions together with ABI3 to regulate genes required for seed maturation and desiccation tolerance. LEC2 regulates synthesis of storage proteins along with FUS3 and LEC1. Hence, the LAFL network regulates diverse seed-specific processes, including deposition of storage reserves, acquisition of desiccation tolerance, developmental arrest of the embryo, and the induction of dormancy. Direct targets of the LAFL transcription factors include seed storage protein genes, late embryogenesis or LEA genes, which protect proteins against desiccation stress, and genes that regulate the expression of ABA biosynthesis and signaling components. The LAFL network also represses genes that are required for the transition from embryonic to vegetative development, thus preventing precocious germination. Let us now study the reserve products accumulated in mature seeds and how the seeds acquire desiccation tolerance during maturation. Both the endosperm and the cotyledons can accumulate seed storage compounds during seed maturation. These reserves are important sources of nutrients to the germinating seed till the young seedling attains the ability to carry out photosynthesis. In some seeds like cereals, the endosperm is the main storage tissue of seed reserves. In others, the endosperm is short-lived and undergoes programmed cell death during development, making cotyledons the main storage organs. Seeds are known to store nutrient reserves in the form of carbohydrates, proteins, or lipids, which facilitate the growth of seedling after germination. Storage starch in developing seeds is synthesized in amyloplasts, which are present in the endosperm or cotyledons. The raffinose family oligosaccharides or RFOs, which include raffinose and stachyose, are oligosaccharides that are storage carbohydrates in some types of seeds. Lipids in the form of triacylglycerols are stored in distinct membrane-bound organelles called lipid bodies that are present in the cells of storage tissues. The triacylglycerols are synthesized by the binding of fatty acids to glycerol. Seed storage proteins are synthesized during seed development within the endosperm or cotyledon cells. Legumes store proteins that belong to a class called globulins. Cereals show two classes of storage proteins, the water-soluble globulins and the more hydrophobic prolamins. Prolamins and globulins accumulate in storage vacuoles which are transformed into protein bodies as more and more protein accumulates within them. The accumulation of seed storage reserves is stimulated by abscisic acid, and many of the seed storage protein genes have an abscisic acid response element in their promoters. Packing of insoluble seed reserves in the cells of the endosperm or cotyledons leads to a decrease in their water content, and this marks the beginning of the desiccation phase in the seed. At the end of seed filling phase, most of the endosperm and cotyledonary cells are dead, though they remain intact till they are depleted of their reserves during germination. The embryo axis is, however, living and needs to be protected against desiccation-induced damage. The late embryogenesis abundant proteins are a diverse family of proteins expressed during this time, and many of them are ABA responsive. They act by buffering the water loss and maintaining cellular stability within the desiccated seed. However, the mechanism of their action remains largely unknown. Besides LEA proteins, Small cytosolic heat shock proteins or HSPs are also expressed during seed maturation. They function as molecular chaperones by preventing the aggregation of proteins under desiccation stress and facilitating their subsequent reactivation during germination. The transcription factor ABI3 has been shown to play a role in the regulation of HSP gene expression in seeds. After having seen how storage products are accumulated in seeds and how the seeds prevent desiccation tolerance by the expression of proteins like Lea proteins and the heat shock proteins, we now move on to study the development of an important organ in a seed called the endosperm. Successful seed formation requires the simultaneous development of the diploid embryo and the triploid endosperm. The fertilized central cell forms the endosperm, and a major event in endosperm development is the transition between the syncytial phase, during which the endosperm nuclei undergo many rounds of mitosis without cytokinesis to form a syncytium. This is followed by the cellularization phase, during which cell walls are formed around the endosperm nuclei. Regulation of these states of the endosperm is carried out by a transcription factor AGL62, which suppresses the cellularization during the syncytial phase of endosperm formation. During seed development, AGL62 is expressed exclusively in the endosperm, and its expression declines abruptly just before cellularization. On the other hand, another group of genes called the fertilization independent seed or the FIS class of polycomb group genes triggers endosperm cellularization. Now these polycomb group of proteins modify the target genes at the chromatin level through histone modification. It is also involved in preventing the formation of the seed in absence of fertilization through a process called imprinting, which we shall see shortly. Several receptor-like kinases have been shown to be required for proper endosperm and/or embryo development. HICU2 represents one such receptor kinase. IQ encodes a leucine-rich receptor-like kinase that is expressed in the endosperm of developing seeds. IQ mutants show precocious endosperm cellularization as well as decreased endosperm size. Now these RLKs, they require an extracellular signal to trigger their activity. And several polypeptide signaling molecules are involved in regulating seed formation. One such group of signaling molecules are the clavata-related molecules. We have already seen that clavata-3 is the ligand which activates the receptor-like kinase clavata-1 during the formation of early embryogenesis. During endosperm formation, a similar clavata 3 like protein, it is called clavata 3 embryo surrounding region related or the CLE family of proteins. Now one such CLE gene is expressed in early embryos and endosperm. It is the CLE8 gene. CLE8 is involved in patterning of the embryo basal domain, in elongation and patterning of the suspensor cell, and also in endosperm maturation. By regulating both endosperm proliferation and the timing of endosperm differentiation, CLE8 ultimately determines the seed size. Mutant CLE8 seeds are smaller than the wild-type seeds, even when the embryos are morphologically normal. CLE8 has been shown to positively regulate the expression of VOX8, which is a master regulator of early embryogenesis, controlling suspensor development and root initiation in the basal lineage of the embryo. VOX8 is also expressed in the endosperm and is a target of the CLE8 function. Since VOX genes are also involved in promoting cell division rather than differentiation, VOX8 is likely to play a role in promoting endosperm proliferation and thus the seed size. Endosperm proliferation is also regulated by the seed coat, a transcription factor called transparent testa glabra or TTG2, which is expressed in the integuments, promotes cell elongation of the seed coat cells. This allows the endosperm proliferation and an increase in seed size. The TTG2 transcription factor is also known to regulate the production of mucilage and tannin in seed coats of Arabidopsis. In addition, AGL62, which we have just mentioned earlier, is also known to activate auxin synthesis in the endosperm, which later stimulates the development of seed coat and the fruit, both of which are maternal tissues. Hence, the expression of AGL62 in the endosperm provides a mechanism to link fertilization to the development of maternal tissues that play a role in protecting and disseminating the seeds. Finally, we come to the concept of imprinting. An offspring inherits two alleles, one from each parent. For most genes, both the alleles are active in the offspring, but for some genes, one of the alleles is not expressed in the offspring and is said to be imprinted or silenced. Imprinting, therefore, is thought to be a mechanism for balancing the contribution of both parents in the offspring. Imbalance between paternally and maternally inherited genes, for instance, as a result of interploidy crosses, results in aberrant seed development, as we all know. It is predominantly the endosperm and to a far lesser extent the embryo that is affected by such imbalance. Deviation from the normal 2 maternal is to 1 paternal ratio in the endosperm genome has a severe effect on endosperm development and often leads to seed abortion. Now, a theory has been proposed to account for this process of imprinting. According to the parental conflict theory, as it is called, imprinting may have arisen from a conflict between the maternal and paternal genomes in relation to the transfer of nutrients from the mother to the embryo via the endosperm. According to this theory, a gene that tends to suppress endosperm development and restricts nutrient flow to the embryo would be preferentially expressed by the maternal allele, whereas the paternal allele would be silenced. By contrast, a gene that tends to promote endosperm development and therefore increases nutrient flow to the embryo would be preferentially expressed by the paternal allele and the maternal allele would be silenced. Now, the theory also goes on to suppose that since the progeny consists of several fertilization events by diverse paternal parents, the maternal parents, through imprinting, tries to restrict nutrient flow to the embryo from the endosperm in an attempt to distribute the nutrient to maximum number of seeds, thus creating a diverse and large progeny. On the other hand, the paternal parent tries to ensure that the nutrients are preferably transferred via the endosperm to the progeny of that specific paternal parent. About 100 imprinted genes are known in Arabidopsis and a majority are expressed in the endosperm. Let us now see the mechanism that leads to imprinting or silencing of specific genes of the maternal or paternal genomes. The mechanism of gene imprinting involves DNA modifications that is methylation and demethylation of DNA. DNA methylation is carried out by the enzyme methyltransferase or MET1, whereas demethylation of these methylated sites is carried out by a DNA glycosylase called Demeter. The histone modifications are brought about by the polycomb group of proteins, which consists of several members, three of which are shown here. One is the fertilization independent seed or FIS2, media or MEA, and flowering vaginigen or FWA. Now, if you look at the figure on the right, you can see the silencing of these three polycomb group of genes in the sperm cell as well as the paternal genome in the central cell. So these genes which come from the paternal parent are methylated by this MET1 DNA methyltransferase. On the other hand, Demeter demethylates or prevents methylation of the female alleles of these genes. The maternal alleles of the MIA, FIS2 and FWA genes are therefore allowed to be expressed in the endosperm. On the other hand, there is a gene called as FERS1, which is a transcription factor. This transcription factor is seen to be a master regulator of several paternally expressed imprinted genes in the endosperm. So what does this mean? FERS1 shows DNA methylation on the maternal alleles and the paternal alleles are expressed. Several genes that are required for endosperm proliferation and growth are under the transcriptional control of FERS1. As FERS1 is paternally expressed, therefore, proliferation of the endosperm becomes dependent on the presence of the paternal genome in the endosperm, which allows fertilization event to be coupled with the onset of endosperm growth. To summarize, after completion of morphogenesis in which the body plan is laid out, the embryo starts accumulating storage compounds. It acquires low humidity levels and desiccation tolerance and enters a dormant stage. The end of seed maturation is characterized by decreased gene expression and metabolic activity. The dry seed, therefore, represents a pause in the plant's life cycle, which can withstand unfavorable environmental conditions. Seeds are dispersed at this stage, and under favorable conditions, they germinate to form a plant. In the next module, we shall see how the states of dormancy and germination are regulated.